Cell respiration is the controlled release of energy from organic compounds to produce ATP. This process supplies energy for the functions of life. Cell respiration can be divided into two major categories: aerobic respiration and anaerobic respiration. Aerobic respiration requires oxygen and, compared to anaerobic respiration, gives a large yield of ATP from the oxidation of glucose. The overall process of cell respiration can be outlined by the following equation. C6H12O6 plus 6O2 yields 6CO2 plus 6H2O plus ATP, or glucose plus 6 oxygens yield 6 carbon dioxides and 6 waters plus ATP. However, cells must go through many steps before these end products of 6CO2, 6H2O, and ATP can be reached. There are 4 stages that make up the process of aerobic respiration. They are, in this order, glycolysis, the link reaction, the Krebs cycle, and chemiosmosis. Glycolysis, the first stage of cell respiration, occurs in the cell's cytoplasm. During glycolysis, a 6-carbon sugar, a hexosugar, glucose, is phosphorylated. During phosphorylation, phosphate molecules are added to another molecule. ATP helps with this phosphorylation of the hexosugar, but for ATP to help, it is facilitated by the enzyme hexokinase. Once the ATP is used, it becomes ADP. Phosphorylation occurs twice to the glucose molecule, one phosphate group being added each time. The 6-carbon molecule is now unstable. This molecule splits into 2 triose phosphates, 2 3-carbon molecules, as a result of this instability. Next, NAD is oxidized, becoming NADH. The energy release from the hydrogen molecules allows for the 2 ATP to form. 4 ATP are produced, but there is only a net gain of 2 ATP since ATP was used at the beginning of glycolysis for phosphorylation. Additionally, 2 pyruvate molecules are produced as a result of the 2 triose phosphates oxidizing. These 2 pyruvate molecules are the end product of glycolysis. Glycolysis is followed by the link reaction in anaerobic respiration. During the link reaction, the pyruvate formed from glycolysis enters the mitochondria matrix from the cytoplasm. Pyruvate is decarboxylated in oxidative decarboxylation. This means that it's split and oxidized when enzymes remove 1 CO2 and H from pyruvate. NAD then accepts the H to form NADH. This converts pyruvate into an acetyl compound, which then attaches to coenzyme A. This forms acetyl-CoA, which moves on to the next stage of aerobic respiration. The Krebs cycle follows the link reaction. The Krebs cycle also occurs in the matrix. The first step to occur in the Krebs cycle is the joining of the 2-carbon acetyl group to a 4-carbon molecule. This reaction is facilitated by the enzyme that helps make up acetyl-CoA. Once the resulting 6-carbon molecule is formed, the enzyme leaves it and returns back to the link reaction. Then the 6-carbon molecule undergoes oxidative decarboxylation. This releases a molecule of carbon dioxide and NAD becomes NADH, which goes to the electron transport chain, or ETC. Now the 6-carbon molecule, which is called citrate, is a 5-carbon molecule. This molecule undergoes oxidative decarboxylation again. Another carbon dioxide molecule is released and another NAD becomes NADH. The 5-carbon molecule is now a 4-carbon molecule. This 4-carbon molecule goes through rearrangement as ADP becomes ATP. FAD, which like NAD is an electron carrier, becomes FADH2, and NAD becomes NADH. FADH2 and NADH go to the electron transport chain following the 4 molecules rearrangement. The net yield of products per glucose that undergo the Krebs cycle is 2 ATP, 6 NADH plus a proton, and 2 FADH2. This is the case because each of the 2 pyruvates form 2 acetyl-CoAs during the link reaction. So each acetyl-CoA goes through the Krebs cycle. Following the Krebs cycle is chemiosmosis. Chemiosmosis occurs across the matrix and inner membrane space through the electron transport chain. Oxidative phosphorylation occurs across the inner mitochondrial membrane. NADH, produced during the Krebs cycle, deposits its electron in the inner membrane and its proton across the membrane in the intermembrane space. NADH is now NAD. Then FADH2 deposits 2 electrons in the intermembrane space and 2 protons across the intermembrane space via the second of the 4 integral proteins in the membrane. The electrons in the inner membrane continue to pass through the integral membrane proteins. Because of all the protons released into the intermembrane space by the 6 NADH and 2 FADH2s produced during the Krebs cycle, there is a high proton gradient in the intermembrane space. Protons diffuse through the 4th integral membrane protein with ATP synthase ending up in the matrix. The incoming protons join with ADP as they come through to generate ATP. The yield of ATP in chemiosmosis is potentially 32, but in most conditions, the yield is slightly lower. For chemiosmosis to keep going, and thus for cell respiration to continue, the hydrogen gradient needs to be maintained. In order to maintain this proton gradient in the intermembrane space, the concentration of hydrogen ions in the matrix is lowered. O2 splits and binds with free protons. Each of the 2 oxygens uses 2 protons and 2 electrons to form water. Thus, 2 water molecules are formed per O2 molecule. Oxygen not only helps maintain the hydrogen gradient, it also helps allow the electron transport chain to continue. It does so by acting as the terminal electron acceptor, and since electrons need to be removed from the electron transport chain for it to continue, the electrons that leave the chain can be accepted by the splitting of the O2 molecule to help create the 2 molecules of water. Chemiosmosis is the last stage of aerobic cell respiration. So after this, the products of 6CO2, 6H2O, and ATP have been produced, and it can start over again with glycolysis. The second category of cell respiration is anaerobic respiration. This type of cell respiration does not require oxygen. Additionally, there are only 2 stages in anaerobic cell respiration: glycolysis and fermentation. The process of glycolysis is the same as it is in aerobic cell respiration. However, instead of the 2 pyruvate molecules produced going on to the link reaction, they go on to fermentation. In animals, lactate is produced during fermentation. In yeast, though, CO2 is released and ethanol is produced. In both cases, the 2 NADH used in these processes revert to 2 NADs afterward. The ethanol from anaerobic respiration in yeast is later used to produce beer, wine, bread, cheese, yogurt, or soy sauce.